Hello everyone, and welcome to today's webinar, Cryogenic Electron Microscopy of Beam and Air-Sensitive Materials. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Daniel Long, postdoctoral appointee and electron microscopist at Sandia National Laboratories, and Dr. John Watt, scientist and electron microscopist, Center for Integrated Nanotechnologies, or CINT, at Los Alamos National Laboratory. Daniel and John, you may now begin your presentation.
Thank you very much for the introduction. Again, my name is Daniel Long. I'm from Sandia National Lab at the Center for Integrated Nanotechnologies. Today I'll be talking about cryogenic electron microscopy of beam and air-sensitive materials as it relates to generally material science. And today's talk will be split in half, myself, and then the other half will be done by John Watt, my colleague from Los Alamos National Lab. Brief outline of today's talk. I'll talk about the benefits of cryogenic electron microscopy as it relates to material science. Then I'll give a short introduction to preparing a liquid-solid interface for cryo-FIB and cryo-TEM investigation. Then I'll talk specifically about a TEM experiment, a TEM investigation of calcium metal anode batteries that I've been involved in recently. And then once those three topics are finished, John will talk about cryo-EM of soft matter. So the benefits of cryo-electron microscopy are many. I've listed quite a few on the right. That's not fully exhaustive. But when it pertains to material science, it's especially important for sample preparation and sample imaging. You can see on the left here, this is a FIB cross-section of a lithium metal substrate. You can see at room temperature, the FIB cross-section has a lot of remnant damage, whereas at cryogenic temperatures, it's nice and clean. So there's a lot less ion beam damage with when you cool the sample. When it comes to electron beam damage, say in the TEM, let's say we're looking at lithium dendrites, lithium metal dendrites. You can see in the top left image that is a slightly corroded lithium metal dendrite that has holes in it that formed during high resolution imaging. Whereas on the bottom, the lithium dendrite is fully metallic throughout the thickness of the sample, and you can image it to atomic resolution fairly readily. So it's really nice for that. It's also great for reducing ion beam artifacts, such as alloying, embrittlement, or hydrogenation. And here's an example on the left of an aluminum grain boundary, looking at the chemical composition across that grain boundary. And on the left part of the figure, you can see that that gallium has implanted itself between these two grains. On the right side of the figure, the gallium has not preferentially embedded itself within the grain boundary. So it's very useful for those kinds of studies. Also great for stabilizing organics, nanoparticles, those sorts of things. So here's a fracture cross-section of a hydrated microgel. This is kind of similar to the work that John will be showing at his half of the talk. And then one of the most important things that cryo cryogenic electron microscopy allows us to do is to look at liquid-solid interfaces. And when I say liquid-solid, I mean a vitrified liquid and against a solid interface. And here's an example of some of my work where I looked at a surfactant solution against a mica substrate, looking at how the surfactant solution behaves at that substrate. These are great for, especially for batteries, modern batteries that have liquid solvents, liquid electrolytes, other devices with trapped or flowing liquid, as well as liquid storage. And a little bit more flavor of my work, I primarily work in the energy storage areas. So I've looked at single-use lithium-ion battery anodes and cathodes. I've looked at lithium metal anode batteries on the bottom left. I've looked at multivalent metal anode batteries, specifically calcium and magnesium on the top right. And then I've also looked at the aqueous mica interfaces. So how do we prepare these interfaces? It's a complicated process, and I broke it down into 10 steps here. And it can be broken down in various different ways. But the first step is vitrification of your liquid-solid interface. And I have two examples. The top example is showing a circular calcium deposit on a gold-coated silicon substrate. And those are generally vitrified in liquid nitrogen. The higher molecular weight of the electrolytes allows you to cool the sample more slowly and still vitrify the electrolyte. Whereas on the bottom, I have a image of a mica H2O sample setup, and those need to be vitrified in a vitribot or in liquid ethane. Then the samples are transferred into our Leica cryogenic suite, and that involves our load station, which is underneath the title there. And that load station has a shuttle attached on the left side, and we use that shuttle to move the sample between the load station, our sputter coating station, which is on the bottom right, and then our FIB-SEM, which is on the top right there. Third step is to protect the lithium. I'm assuming that, this is assuming that the the liquid is the top layer of your sample. And so the way that this is done in cryogenic FIB-SEM is by a blanket coating. And so the sample is fully cooled to cryogenic temperatures. The gas injection system needle is retracted several millimeters, and the set point is changed to room temperature. And then it's open, the flow is open for on the order of 10 seconds to a minute, and then it's cured with the ion beam. Now this is a black box solution. It's not as good as the room temperature FIB platinum coating. So you do have to tune the parameters so that you get well cured and fully dense platinum coating. And this is great for protecting that liquid layer on top, because that liquid layer is generally going to be the most beam sensitive component of your sample. Moving on to steps 4, 5, and 6, you need to locate your region of interest. Now, when it's coated with a vitrified liquid, typically your surface is not going to have a lot of features. As you can see on the left image, in the top right part of the left image, it's mostly flat with a few features kind of poking through, and so it's difficult to locate where you are in the sample. So I recommend correlating the SEM images to an optical image or a fluorescence image that you've taken prior to going into the SEM. And then 5 and 6, I bunched together. I call that just standard FIB milling. So you do your trenches, kind of your cleaning up the trenches, preparing for the J-cut of the FIB lift out. And this process is highly sample dependent. It will depend on the density of your solid phase, the density of your liquid phase, and how much, and the quality of your FIB platinum cap on top. So before you do the J-cut, you need to protect the backside of the lamella. Assuming that you have a liquid phase present, that liquid phase will damage very easily when you've tilted the sample to tilted the stage to 0 degrees. So what I recommend doing is doing a reduced area in the yellow dashed box area, and just letting that run for a minute or two, and that will redeposit a few hundred nanometers of material onto the back. And this is important for when you do the J-cut, you have to image that backside. Then the next step is attaching the needle. And I've shown here a redeposition attachment. And so you can see the needle is on the right there, on the right image. It's the bottom left part of the image is the needle. And then the sample is the majority of the middle of the image. And there's lines kind of connecting the two parts. And the way that that's done is via a little bit of a complicated process. But if you can imagine visualizing the lift-out needle on the left there, you have your lamella and your new or your unprepared lift-out needle. Generally, they're round when you buy them or you install them. And so what we need to do is to flatten the front side of that lamella needle before making contact with it. And then once that's flattened, you can go ahead and make contact. And then I use thin rectangular patterns that I mill through the needle and then into the sample, and that creates a redeposition connection. And I'll move those patterns sequentially outward, as you can see here. And then I've included a video of this process. So I'll go ahead and talk over this video a little bit. So I'm moving the needle into position. This is my sample, my region of interest already chosen. My trenches are cut. My J-cut is cut. So I'm putting my little patterns there. I'm going to bring the needle into contact. And this is sped up by 10x. I'm not the fast. So I'll let those two patterns move, and then I'll move them outwards, taking sequentially taking images of the connection with the E-beam, and doing a little bit of an extra pattern, and then there's my connection. Then I'll cut the rest of the J-cut, and the sample's completely free, and then I'll lift it out. And so this is a great method for doing cryo lift out. You can also use you can put down more FIB platinum. It does charge if you don't cure it, so that's one issue to think about. You can also put down ice if your FIB system has that available, but again, that will also charge, so I like to do this redeposition cut. And once you have it onto the needle, the next step is to put it onto the FIB grid. So the FIB grids that we're using are a copper FIB half grid that's attached to a molybdenum slot grid, and that molybdenum slot grid gives us a good handle to manipulate the sample when we're loading it under liquid nitrogen. So I'm bringing the sample into contact, made contact. I like to spin the needle till it's almost nothing left right before I make contact. And then I'll use a regular cross section with the beginning of the pattern, so the bottoms of these patterns towards the sample, and that redeposits material backwards into that, and then I'll cut the needle free. And then once I have that needle free, I'll go to 52 degrees, and I will anchor down the other side. And generally, when I do the lamella anchors, I kind of do an angular distribution around the side. And what I'm looking for is to see that kind of the bottom of the sample start to flare outward, indicating that I've got lots of redeposited material there. Okay, so then once that's done, and I'm happy with how rigid my sample is connected to the grid, then I would move on to final thinning. This is one method of attachment. This is on top of a FIB post. You can also attach in the V-notch of your FIB grid if you prefer that. And here's just some other images of me doing that. You can also do it in the flagpole position as well. And it's the same general principle that you use the redeposition to make the connection. So then the last step is final thinning. This is highly sample dependent. I've shown 3 images kind of progress through one of my thinning procedures. And you can see the contrast of each layer changes throughout the process. And so it's very important that you understand how the contrast changes with your material, especially if you have something like an electrolyte that's very low Z, or an organic that's low Z, or you're working with lithium or something like that. And in general, I like to use the low voltages, the ion voltages of 8, 5, and 2 a lot more than I traditionally do in room temperature prep. And I can generally get these samples to be about 50 nanometers thick pretty much every time I get a good lift out done. Okay, so then we're going to switch gears. So now that I've got that kind of laid out for you, I'll talk about one of the research projects that I'm involved with, which is, again, calcium metal anodes. And this work was funded by JCESR, that's the Joint Center for Energy Storage Research. And this is in collaboration with Scott McClary and Kevin Zabadil. So briefly, lithium-ion batteries are the current kind of dominant energy storage, especially mobile energy storage. They're typically composed of an anode that is coated with graphite, and then the cathode is coated with transition metal oxide, lithium cobalt oxide, or other alloys of that. And then they're separated by a polymer separator, and then that's infilled with the liquid electrolyte. However, as we continue to increase our energy demands, say from mobile devices to cars to home storage to grid storage, we have kind of a fundamental issue with continuing to use lithium batteries, and that is that lithium batteries with their carbon-based anodes have a reasonable but small energy capacity. And especially in addition to that, they also have a low natural abundance compared to some of these rock-forming elements. So when we look at these rock-forming elements, things like calcium, aluminum, sodium, and magnesium all have much higher natural abundances. And then if you go to the middle, you can also see that their theoretical volumetric capacities are also much greater than current lithium-ion technology. So we have to ask ourselves how far we can really take these lithium-ion batteries. So recently, I think it was 2018, there was the first publication of a room temperature cyclable calcium metal anode battery. And since then, there's been a whole bunch of publications that have followed along after that. And these are all great, that this is possible and that we can study the system now, and we can include this in our kind of our research repertoire. But we also need to realize that we need very high coulombic efficiencies, greater than 99%, for practical rechargeable batteries. And these rechargeable batteries require a very ideal type of SEI, or solid electrolyte interface, and a very ideal deposit morphology. We don't want anything that's going to grow dendrites or grow in a very structured manner that could puncture a battery. And so here's what that ideal battery might look like. It'd have a continuous SEI that would only allow for the metal to transport across. It wouldn't allow for any solvent or anion transport, and it would not allow for electron transport either. And then that ideal battery would also have a fully dense, if we're talking about metal anode batteries, a fully dense metal anode that is relatively flat and well behaved. So that first paper that I showed, they did some SEM on their calcium deposits, and they found what they believed was a calcium hydride phase in their SEI. And that's okay, that's pretty good, because another group modeled the migration barriers of both calcium and magnesium metal anode SEI component. And they found, if you look on the left there, MH2, that's the metal hydride, that they have very low migration energy barriers, which is great, great news for an SEI component. However, this was a few years old, and this experiment on the right here is a few years old, and they didn't necessarily use really inert methods to look at this. And so we need to restudy, reinvestigate this material system, this electrolyte that they specifically used as well with cryogenic and inert methods. So briefly, we used Teflon T cells with a calcium counter and reference electrode, and we deposited onto gold textured working electrodes. Our electrolyte was the same as the paper that we previously showed, which is a calcium borohydride salt with a little bit of sodium borohydrate salt that are both solvated in THF solvents. And looking at plan view of these deposits, they look pretty dense. There's a few pockets of porosity, but for the most part, they're completely dense. We deposit them to a few hundred nanometers thickness just because we're interested in early stages of growth and nucleation. XRD confirms what the other authors have found, that they have primarily HCP and FCC phases of calcium metal, and we had trace amounts of calcium hydride as well. And so this is all kind of gelling together into one nice story. However, XRD is not spatially resolved here. So we need to understand how that works. That's why we included cryo-FIB and cryo-TEM into these efforts. So I'm going to skip through all the cryo-FIB preparation and jump right into the TEM results. So on the left is a bright field image of a cryo-FIB lift-out lamella of these battery systems, and you can see that the deposit is completely dense. We do have electrolyte present at the top of the image. It's a little bit cut off here, but there is electrolyte present as well. It's fairly textured. You can see kind of linear features on the right side going from the gold electrode to the top. And if we look at a selected area diffraction pattern on the right, we have lots of rings that when we index, they index to HCP and FCC calcium, so this is all consistent. And there's also a diffuse calcium oxide ring hidden in there as well, which we believe is on the front and back of the lamella, and that's just some trace oxidation. But what this diffraction pattern tells us is that, one, we have a very textured deposit, which is to be expected with a textured electrode, but we also have a lot of stacking faults, and that's evident in the streaking in the diffraction pattern. And that's to be expected when you have a dual FCC-HCP phase system. When we look at this with STEM, something pops out to us. We see these nuggets of interesting contrast, this different porosity near the gold electrode. And when we take a selected area diffraction pattern from those regions specifically, we find calcium hydride reflection. And so what this is telling us is that calcium hydride, at least the majority of what we're finding, is located near the gold electrode. So that leads us to our next investigation, which is a chemical investigation using electron energy loss spectroscopy to look at the chemical fingerprints across this deposit. So we looked at this region of interest. That includes the hydride, which I've outlined here in yellow. And what we did is we did low loss EELS mapping, and then we applied principal component analysis and pulled out the principal components and then mapped them. And what we found was we have 4 principal components. We have the metal component, hydride component, electrolyte component, and the SEI. You can notice that the SEI and the hydride are distinctly different. They do have similarities, but they are distinctly different. When we map those components, what we see is that the SEI, which is located where you'd expect it on the surface of the metal, mapped as green here, is not showing up in the hydride, and the hydride is not showing up in the SEI. When we move to pore loss EELS, a similar story evolves. This time we have 5 principal components. That's just because the electrolyte split off into a carbon-rich and a boron-rich component. When we map them, same story, same picture arises. Hydride is distinctly different than the SEI. And these are mapped at 10 nanometers per pixel. We do have higher spatial resolution maps as well that confirm this. When we zoom into the oxygen edge of the top 3 components, seeing the green component, the SEI, that it's oxygen rich compared to the other two. And so this is kind of pointing us in the direction that this is a calcium oxide phase that's present. So when we image this, it's probably hard to see on your screens here, but I'll go ahead and outline it. We divided this image into metallic calcium at the top and interphase region in our electrolyte region. And zooming in on little spots in metallic calcium, we have HCP as well as FCC that I didn't show, lattice spacings. In the interphase, we have lots of oxide, lots of oxide lattice fringes. And so this is pretty strong evidence that both the SEI is both structurally heterogeneous because we have these different orientations, and it's confirmed to be calcium oxide, likely with some carbon and some boron present. So where that leaves us is a picture like this. We have only calcium hydride domains near the gold electrode, which is okay because I'll show you in a second. The heterogeneous calcium oxide also has a somewhat manageable migration barrier. You can see here the MO, it's the red MO. It is higher than the hydride, but it's not as high as the magnesium case. And so these are ongoing research studies that we're doing, and we will be publishing this briefly. And then with that, I'd like to acknowledge again Scott for sample generation and the electrochemistry, and Paul Catula for helping with data collection and data analysis, and also thank Jay Caesar for funding this work. And with that, I will turn it over to John.
All right. Thank you, Dan. Thank you for first half of the talk. So my name is John Watt. I'm also at the Center for Integrated Nanotechnologies. I'm also at Los Alamos National Laboratory. So I'm going to shift gears here a little bit and talk about cryogenic electron microscopy for soft matter materials. So obviously cryo-EM has seen a huge explosion recently, mostly in structural biology, where they're really pushing the resolution limits, for structural biology and solving the structures of proteins. Here is a very high resolution image of the COVID spike protein. So here the goal is, along with most TEM, is to increase resolution, and they do this with very high accelerating voltages and aberration correction. But unfortunately, when you do that, you drastically reduce contrast. This is an issue with these types of low Z samples, as you need some contrast to see things. So to counteract this, these are suspended in a vitreous ice background, also playing with things like defocus and phase plates, and also taking many hundreds and thousands of images of the protein. So on the right there, you can see, a typical kind of single image of a protein sample in cryo-EM. Each individual structure is fairly difficult to see with fairly low contrast, but you reconstruct thousands of those images together, and all of a sudden you get your structure. At CINT, at the Center for Integrated Nanotechnologies, we're really interested in material science and nanotechnology. So we are using cryo-EM for a different sample space, and that's soft matter. So things like polymer vesicles, micelles, microtubules, nanoparticles, nanoparticle polymer composite, even things like metal organic framework. So soft matter has historically seen limited analysis by transmission electron microscopy due to a few inherent problems. One is that it gets damaged by the electron beam. It's a soft matter. It also has an instability under a high vacuum environment. This is especially true if it's hydrated or solvated. To look at it, you typically have to drop cast onto a grid and then dry off the solvent. And that gives you a lot of drying artifacts and uncertainty in your structure. to counteract the low contrast of, carbon against the carbon film background. Traditionally, folks will use negative staining techniques. There's something like urinal acetate, and it's an image, typical image on the top right there. So you get good contrast, but here again you have drying artifacts, and also now you're putting a very heavy metal into your sample, and so it introduces more uncertainty into the structure. So analysis of soft matter has been dominated by ensemble scattering techniques. So two of the main ones are dynamic light scattering, DLS, and small angle X-ray scattering, stacks. So two examples here in the graph. So these are very useful, but they're also, they average across the entire ensemble. So they don't give you a real space image, and also, they're based upon a model. So we really want to see a real space image that doesn't have all of these drying artifacts and staining artifacts in soft matter research. So just a few examples of synthetic organic nanostructures from the literature and what they look like in cryo-EM. So on the left there, we have liposomes with citrate stabilized gold nanoparticles, and you can see the gold nanoparticles have very good contrast, as we would expect. And then looking at the liposome, if you look at it against the vitreous ice background, so if we look at frame F, you can see against the vitreous ice we have good contrast. But as the structure kind of overlaps with the carbon substrate, you see we lose that contrast. So really keeping these in the vitreous ice is our best chance of seeing the structure. Another couple of examples, cross-linked nanobubbles of a perfluorocarbon, and then also, nanoparticle polymer conjugates. So these are polyacrylic acid, polystyrene, brush-grafted silicon nanoparticles. Some of the examples from CINT is quantum dot polymer nanocomposites. So these are from Stacey Kopp out at UC Irvine. So we can see, the structure of the quantum dots within this nanocomposite. And then also against the vitreous ice, we can see the polymer and make out the shell there. On the right-hand side, we have silicon nanoparticles that have a lipid coating. So this is Professor Nuruddin from University of New Mexico. And when we use low accelerating voltage, high contrast imaging, we can resolve at least the visible lipid on the surface. And so, these kinds of things are really unique to cryo-EM. If we were to put this into a high accelerating voltage dry TEM, we just wouldn't see that lipid layer. So the CINT cryo-EM suite, and Dan kind of touched on a few of these tools, but I'll just give an overview as to what we have and some of the unique capability. So this is a microscopy suite dedicated to minimizing electron dose for the imaging of materials and their interfaces and their native hydrated or solvated state. We've actually recently done some experiments with toluene, freezing toluene, providing our user community with optimized characterization of sensitive materials across multiple length scales. And it's actually the Leica equipment that really helps us to cross those multiple length scales. So within the suite, we have a vitribot. So that's for consistent and reproducible plunge freezing. And we have the SIOS focused ion beam milling SEM. And then we also have a dedicated Talos L120C cryo-TEM. So, this lab has been set up for the analysis of soft matter. And so this Talos is a low accelerating voltage, but very high contrast tool. So it mitigates as much beam damage as possible while giving us really good contrast and resolution. But it's the cryo stage and transfer equipment that really kind of ties the whole suite together. So the cryo-SEM stage goes on the SIOS, and then the transfer equipment enables us to have all the tools brought together. And this gives us imaging capability for biomimetic materials, polymers, nanocomposites, and interfaces, and also, as Dan was presenting, low Z systems and those which so the Leica cryo workflow, it really is a complete and robust workflows. So we have the vitribot, then we have the VCM. So this is where we can, transfer in our frozen samples, and we can manipulate them. We can load them into the shuttle. Then we can put them into the A600, which is a cryogenically cooled sputter coater. And then, as Dan has shown, we can go between the SEM and the TEM with prepared lamella. And that's done on the VCM, where you see the dark spots on the right-hand side, a side port where you can put a side entry TEM holder and then do your transfer completely under LN2. So the nice thing about this system is that once your sample is frozen, it never again sees ambient conditions. You can do all of your work under LN2 or under vacuum, as well as all transfers. So just to briefly go over the kind of steps of plunge freezing. So if you're looking at something like a protein or a vesicle or a nanoparticle that's hydrated and suspended in solution, you would do kind of typical plunge freezing in the vitribot. And so here what you do is you take a wholly carbon grid, which has a carbon substrate, and then some pores. You would apply your sample, and so this is a sample solution, well suspended biomolecules. If you were to freeze that, then obviously you would have a thick layer of ice that the electron beam could not see through. So you wick away the excess liquid with filter paper, and the idea is that the solution will flow into the pores and you will just wick away the excess. You then plunge freeze into ethane, which has very good thermal transport properties, and then you will have a well-suspended sample within that vitreous ice, which is kind of tens of nanometers thick. So just looking at this cross-section, you see that, you do have some D variation in your structure, which you don't necessarily have with dry TEM. So that can, give you some uncertainty in orientation, but also focus. So this is great for something that is, a small particle, well-dispersed in solution. But for bulk materials, if you're preparing for, for FIB, then this cannot always be the best option because, it's quite thick, and also that means it might not hold on to the tweezers, it might not fit with the whole system. And the other thing for polymers that we've found is that they can be very brittle under LN2, and so trying to kind of mount them and manipulate them after they're frozen, you can kind of just shatter them. It can be quite difficult. So one method that we found works pretty well is to mount the sample at room temperature and then just freeze the whole setup. So on the left-hand side there, the brass kind of shuttle at the bottom of the LICO equipment, and then we have a modified FIB setup on the top there with our FIB grid, our FIB half grid mounted, and then an SEM sub with a clamp. And so the idea is that you would prepare this outside and then plunge freeze it into LN2 within the VCM. If it's hydrated, then as long as it's not going to dry out over the course of kind of 20, 30 seconds as you get it ready to plunge, then it's usually okay. And wicking away a little bit of excess moisture with a chem wipe can mean that you have, less ice formation on the surface. So I'll move to the next slide to show these videos in full screen. So here we are just plunge freezing the whole setup into LN2 and waiting for it to stop bubbling. And then this is using the VCT. You'll see the grabber tool come in, grab the shuttle, and pull it back, and then we need to do a little bit of gymnastics with that sample dimensions, and then that goes under vacuum and gets transferred to the SEM. So CINT is a user facility, so some of the most exciting work that we do is with our users. So I'm just going to go over a few examples here of how the cryo suite and the LICA workflow have really impacted user science. So the first example is with Professor Ben Wiley from Duke University. And so this work was looking at an artificial cartilage hydrogel that's strong enough to work in knees. So they had a biomimetic approach to create the first hydrogel with the same strength and modulus as human articular cartilage in compression and tension. The bacterial cellulose nanofiber network with a polyvinyl alcohol and propane sulfonic acid, sodium salt, was formed into a double network hydrogel that exhibited a coefficient of friction about half that of cartilage. It was more resistant to wear than PVA and showed fatigue strength equivalent to cartilage at very high number of cycles. So it was the cellulose that provided tensile strength in a manner analogous to the cartilage, while the PAMPS provided a fixed negative charge, similar to the role of aggregate in cartilage, and it was able to balance a very heavy load. And so we were able to image this on using the cryo-SEM. So this was just freezing in LN2 and then transferring to the SEM. And so here we're seeing the distribution of the fibers without any kind of drying artifacts that would be inherent in normal SEM. Another example, and this is an example of cryo-FIB lift-out and lamella preparation of a polymer. So this is with a CINT user and collaborator from the University of New South Wales, Darway Way. And here they made tungstate doped polypyrrole films with supercapacitors, and they wanted to look at the distribution of tungstate aggregates within the polypyrrole matrix. So here we made really good use of the VCM. This is a very thin film that became quite brittle under LN2, so we mounted it first and then froze it. And then you'll see in Figure A that we made, our FIB cuts as Dan had kind of shown. Polymers can mill very fast. So again, each sample is different, and you have to kind of tune your conditions. Then we did a lift out, and this is the standard kind of flag orientation of the lamella, and then thin. You'll see in panel C that there's quite a bit of ice contamination. So this is actually one of our earliest goes at a polymer lift out, and there was quite a bit of contamination. But we've since got a lot better at doing transfers. Things like heating our nitrogen lines during purge cycles can actually go a long way to reducing condensation in ice. And you'll see in panel D, we do resolve those tungsten aggregates. So, this resolution is on the order of nanometers, which is pretty good for a polymer in a low contrast, in a low accelerating voltage medium. This is kind of, again, repeating what Dan showed, but just showing that for soft matter, for polymers, attaching the needle, the same things ring true. You want very good contact between the needle and the polymer. When you're making your attachment cuts and your redep cuts, you've got to be aware that the polymer mills incredibly fast. So you have to be careful. You've done all that work to make the FIB cuts and bring the needle in. You kind of have to go slow and make sure that you're not completely destroying your sample before you get a chance to attach it. But then we do add a little bit of platinum, cure it, and then lift out. Another thing that's really interesting about cryo-EM is that it can be used on things that aren't necessarily hydrated and don't necessarily need to be kept cold in terms of the sample itself. So this is with collaborator and CINT user Zheng Changyu from Los Alamos National Laboratory. So here they grew some zinc oxide nanorods on a zinc oxide substrate with a 2D molybdenum disulfide interlayer. And the goal here is to preserve that sensitive 2D layer, which when the lift out was being done in room temperature, they were seeing that get destroyed. So on the right-hand side there, you'll see the cut. So this was an interesting challenge because I had to mill from the front to find the rod and then mill from the back so that I could keep that rod intact any further milling from the front meant that I would lose it and I would have to find another one. So this was all performed at, at cryogenic temperatures, minus 150 C. You'll see on the left, when we don't have our milling conditions right, that sensitive interface gets destroyed and you get a void formation. And then on the right, you'll see when we have our milling conditions right, we're at cold temperatures, we're being very careful with the sample, we maintain that sensitive interface. And this is just taking that same sample through scanning transmission electron microscopy. And you'll see on the left, you'll see the platinum, the rod. Also looking at the platinum deposition, that middle layer, you'll see that it's not beautifully uniform because we did get some shadowing from the rod. And so you had to play some tricks to make sure that you got good platinum depth around the rod. And then as we increase the magnification, we can see that we did maintain that sensitive interlayer. So CINT is a user facility. We're funded by the DOE, Department of Energy. So access to our facilities and scientific expertise is free. So access to this cryo-EM lab is free for non-proprietary research. It just requires the submission and acceptance of a 2-page user proposal. So to learn more about the facilities, please visit cint.lao.gov. So we are supported by the DOE, and we are hosted by both Sandia National Laboratories and Los Alamos National Laboratories. And if you had any more questions about the cryo lab, about cryo-FIB lift out, or the analysis of soft matter under cryo-EM, please don't hesitate to contact myself or Dan. I'd also like to thank my colleague Katie Young-Yahan at Sandia National Laboratories.
Thank you, Daniel and John, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is for Daniel. How do you protect the backside again to make the J cut?
That's a good question. So what I do is, with the sample tilted to the ion beam at 52 degrees, I place a pattern on the back side of the sample. That's the side you can't see with the electron beam. I'll place the pattern, and it'll just be a raster pattern or a random pattern, and I'll let it mill on the trench itself, not hitting the sample, but just on the trench itself, and that will redeposit material onto the backside. And that's important because when you do the J cut, you have to image the backside. So if you have a very sensitive sample, you're going to be damaging that backside.
Thank you, Daniel. Let's see, the next question is for John. What are some of the biggest challenges for lift out of polymers and soft matter?
Thanks. Yeah, quite a few actually. I guess as Dan alluded to earlier, for each sample it's very dependent on the nature of the material for things like milling rates, and that's not just for making the cuts, but also things like redeposition. For soft matter, redeposition can be quite a challenge. Also, artifacts from the ion beam into your cuts are quite important. During thinning, you can actually get the lamella curl up on itself maybe a lot quicker than you would for, say, some kind of inorganic material, just because of it's not so rigid. And I would say in cryo, one of the challenges as the sample gets a little bit more sensitive is, Dan showed we have to use a lot of these kind of redeposition techniques for attachment because we can't use, defined platinum welds. That a lot of the redep from soft matter, it's not as controlled, and so you might need to rely on redep from the needle or redep from the platinum protective layer in order to get those connections.
Thank you, John. Our next question is for Daniel again. How long does it take to prepare a FIB lamella of a vitrified liquid-solid interface and also a polymer soft material?
Yeah, that's a good question. So it depends on the size of the lamella that you're making. Generally, I make a fairly large lamella, about 20 to 30 microns wide. That gives me a greater opportunity to get a good region of the sample. But I'd say in general, it'll take you the better part of a full workday to make a cryogenic lamella. Whereas in room temperature, you could do that in a morning or an afternoon.
All right, thank you, Daniel. Let's see, next question is for John. What type of TEM cryo holder and TEM are used? And how about the stability of it? Can you do EELS mapping?
Yeah, that's a great question too, being, kind of the end point of all of this work, right? So you do a lot of work to do the cryo-FIB lift out, you do the transfer, and then, yeah, if your holder's not stable, then it wasn't really worth doing the sample prep. So all of this work was done using the Gatan 626, so kind of the classic cryo holder for an FEI or Thermo Fisher microscope, but really whichever one you have in your lab. So we use the 626. I know Gatan has the ALSA now, which is used more for tomography and has better stability in tomography. There has been some EELS mapping. I think Dan has done some EELS mapping. So yeah, this is all possible just depending on what setup you have for your TEM at the end case. But yeah, absolutely, having confidence in your TEM holder and the transfer is absolutely critical to want to put in a day or 2 days of sample prep.
All right, thank you, John. Next question is for Daniel. Will the cryo experiments on TEM cause contamination or another issue to the TEM?
Another good question. So generally when the TEM holder and your sample are cold, it will act as a contamination trap, and it will, in general, improve the vacuum of the TEM. But if you do have something that sublimates or sputters really easily, something carbonaceous or organic, in theory it could slightly contaminate, but it's not likely. It's more likely to clean up the vacuum.
All right, thank you, Daniel. Our next question is for John. How do you think AI/ML will impact the structural biology?
Yeah, that's quite a heavy question. It's kind of a little bit outside my sphere, but I imagine that will be a huge factor going forward in structural biology, especially when they are trying to push the resolution, more to the sub-angstrom level. And also, I guess the goal would be to, reduce as they start to build these training data sets on, particles that have been resolved to high resolution, you can start to reduce some of the acquisition times that are needed, tens of thousands of images that are combined. Perhaps AI/ML can start to reduce, the time with the microscope, which is obviously labor intensive and, microscope intensive and costly.
All right, thank you. Next question here is for Daniel. Is there advice on ice contamination during transfer to the TEM for battery materials after FIB? And how long do you take out the sample from FIB and bring it to the TEM?
Yeah, so that is a difficult part of the process. It's the one that we had to spend a lot of time in our process development on. But in general, my advice for transfer between FIB and TEM is to keep it under liquid nitrogen, if that's okay. Keep it under liquid nitrogen for as long as possible, and minimize any of the the nitrogen gas exposures, such as when you load it physically into the tip of the TEM, make those as quick as possible. And these are steps that take a lot of practice, a lot of manual, fine motor skills. And then the other question was how long between the two microscopes. For us, we like to do it the same day if possible, but often what happens is we spend the whole first day of the experiment doing the cryo-FIB and lift out and thinning, and then we'll store it in liquid nitrogen in a, I think it's a 35-liter liquid nitrogen storage dewar overnight. And so between generation and analysis, sometimes it's up to 24 hours.
All right, thank you, Daniel. Next question is for John. How do you transfer the air-sensitive sample to the TEM?
Yeah, that's a good question too. That's a very critical step. So I can't recall, Dan, if in your slides you showed the glove box attachment, but what we also have is the VCT, the sample transfer unit. We have a flange on the side of the glove box. So traditionally, if people are trying to transfer air-sensitive samples, they will put it in basically in a mason jar and then put that in a bag and then, run that down the hall, quickly open it up and pump down the SEM with minimal exposure. But we can put the VCT directly on the flange, which goes into the glove box. So we can do things like open coin cells under argon and then transfer them under vacuum and cold. So, it can be under high vacuum and then we can cool it down even more to increase that vacuum and then put that onto the SEM. So that's one of the really nice things about the Leica system is, your sample can never ever see ambient conditions, which is really nice.
Thank you, John. We have one last question here, and it is for Daniel. For one cryo-TEM eels work, how long does it take usually? Double the normal stem eels? And do you have a cryo-TEM holder that also has an air-free function?
So for cryo eels, the main time constraint is drift correction. Obviously with the TEM holder, the 66 that we use, it drifts at, I think it's about a nanometer per second or a little bit less. But we do have to use heavy drift correction. And that adds, I think it's about 20% of the total acquisition time. So if you have a 25-minute acquisition, it'll add another 5-10 minutes onto it. And then we don't have an inert cryo TEM holder. We just have the cryo, the Gatan 626. And I forget what the other model is, but we have a single tilt and a double tilt. And both are just cryogenic holders. But I know that some other institutions have the vacuum and the cryo capabilities.
I will add on that, though, that, the cryo holder does have the frost shield, which even though it's not, air-free, it does give you, a little bit more time to transfer a sample.
Yeah, absolutely. And we've transferred lithium, calcium, sodium, various reactive metals, and very minimal, if not equivalent to a vacuum transfer holder.
Great. Well, thank you again, Dr. Daniel Long and Dr. John Watt, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.